So chapter 8 is about microbial genetics, and in this chapter we're going to learn how genes are expressed and what is the importance of controlling sometimes this expression. So here we have plasmids that are circular forms of DNA that exist in certain bacteria and that has these genes that encode frequently for resistance against antimicrobials. So we're going to see what is the field of genetics, what is some of these terms like genome, chromosomes, genetic code, genotype, phenotype, etc. And we're going to describe how DNA is the source of information for the cells. And we're going to see the process of translation, transcription. And we're going to compare translation between prokaryotes and eukaryotes. So genetics is a study of inheritance, the study of genes, how these genes carry information and how this information is expressed. And how do you replicate this information? Now, chromosomes will be the physical structures that carry this genetic information. And we have, in the case of prokaryotes, for the most part, we have a single circular chromosome. And then eukaryotes, they have multiple chromosomes that they are linear. Within these chromosomes, we have segments that we call genes. And these genes are encoding information for the cells. So sometimes it's functional information, sometimes it's structural information. So usually the products of gene expression are proteins. And again, these proteins can be structural, or like in the case of channels, or functional, like in the case of enzymes. And then genome will be all the genetic information that is found within a cell. And genomics is the study of these genomes, so includes sequencing and characterizing these genomes. This field, thankfully nowadays, is very advanced. You can have genome sequence within a few hours. And that tells you potentially, for instance, in the case of humans, what diseases you can develop. Okay? And then you can find mutations in these genomes, whether it's bacterial or prokaryotic cells or animal cells. And then you can come up with predictions of what can be the consequences of these mutations. So here is the central dogma. So we already know that DNA, it is transcribed into RNA, and then that transcription can lead to translation of proteins. So that's what we know as forward transcription. Whenever they discovered this in the 1960s, it was kind of a huge deal, and it is good because many people, they were like trying to identify how the transmission of information went between the macromolecules. But then, more recently, we discovered that you can actually make a copy of DNA out of RNA, and this is called reverse transcription. So now, within this DNA, we carry the genetic code, which is the set of rules that determines how a nucleotide sequence is converted to an amino acid sequence. So in DNA, we have a 4-letter alphabet: adenine, cytosine, guanine, and thymine. And then we know that the rule is that adenine binds to thymine, and then that guanosine to cytosine. And then within this DNA, we have segments, genes that are copied to make specifically messenger RNA. So transcription happens in the nucleus, in eukaryotic cells. But in prokaryotic cells happen within the cytoplasm. And then translation in eukaryotic cells happens within the cytoplasm, while in prokaryotes, because they don't have a nuclear compartment, it will happen simultaneously in the cytoplasm. Usually, this DNA will be attached somewhere within the plasma membrane of these prokaryotes. Now, every time that this messenger RNA then is read, and then you produce a protein, you call this expression. So genotype, it is the sequence that we have in genetic information in our DNA. And that genotype varies. It's very, in eukaryotic cells, human eukaryotic cells is very similar, but there are certain variations. And that differences in variations, it is expressed in the phenotype. So the phenotype is everything that is visible. In the case of human cells or humans, that expression, that phenotype can be a specific hair, skin color, eye color, the height, the texture of your hair, if it's straight, if it's curly. So phenotype is the ultimate expression of the genes. So in bacterial cells, you have DNA and associated proteins. So as an example, E. coli has a chromosome that has 4.6 million base pairs. And then you have this super twisted to help to compact this DNA so that these small cells can hold that much information. That's the point of supercoiling your chromosomes. And of course, the same happened for us, for our human cells. We supercoil our chromosomes. And then within this genome, you have short tandem repeats, which are non-coding regions. So these are like 2 to 5 base pair repeats that are non-coding, but that helps to actually space the protein encoding genes. And some of these can be expressed or not, depending on what protein you want to make. And you can process the messenger RNA to make these proteins. So here we have an example of these chromosomes in these bacterial cells. Now, the genetic information can be transferred 2 ways, vertically or horizontally. Vertically is when a bacteria has a specific contact with other bacteria, that it is not part of the progeny. This will be, for instance, when you have an F+ bacteria that has a pilus, and then this bacteria, this F+, has a plasmid DNA, and that plasmid DNA will be transferred into the F- bacteria with this pilus. So that's vertical transfer. Now, horizontal transfer, which also can happen, vertical transfer is I was explaining horizontal transfer. Vertical transfer is when a cell divides by binary fission and then transfer these genes. So that's horizontal and vertical. Now the flow of information then will be, you have a parent cell with the DNA, and then you make a copy of this DNA, and then your daughter cell will be expressing these genes that come from this parent cell through transcription and translation. And if it's like, for instance, the ability to metabolize certain carbohydrates or molecules, then this cell is supposed to be able to do so. If there is no mutations, we are considering non-mutated. So now, so that's expression. That's one way to flow genetic information. Another way is by combining genes. So this is recombination. In some cases, some bacteria can encounter another bacteria. That's when I was telling you about this horizontal transmission. So you have an F minus that perhaps was not utilizing lactose, for instance. And then you have an F plus bacteria that was utilizing lactose. And then when you transfer with this pilus of the F plus to the F minus, this plasmid DNA, then the F minus that didn't utilize lactose before, it starts to utilize lactose because the F plus has transferred the genes for this bacteria to use this lactose. So that's recombination. So you acquire new genes and you make a hybrid. Another one, it is by replication, just plainly passing the genes through binary fission. So that's the flow of genetic information. So what can be a clinical application of genomics?
Searching for like predispositions, like diseases and stuff.
Mm-hmm. Yeah, you can tell the predisposition to specific diseases. And then why is the base pairing of DNA important? Fidelity. So if you always base pair your genes the way that they're supposed to be, there is less risk of mutation. So it's like spelling. We have 27 letters in the alphabet. And then if you always preserve the spelling, that's less ambiguous language. Okay, so for DNA replication, so DNA forms a double helix. You have a backbone where you have this sugar, deoxyribose, and you have a nucleotide attached to this. So the nucleotides face medially of the two strands, and then base pair accordingly, and then you have hydrogen bonds. 3 hydrogen bonds between guanine and cytosine, and 2 between adenines and thymines. Depending on the genome, you can have specific areas where you have more ATs than CGs. And if you're doing PCR, you will take into account that. If you go into a region where you have a bunch of Gs and Cs, your PCR temperature has to go up. Why? Because you have to break down these triple bonds of hydrogen, which are stronger than double bonds. Now, one thing about these two strands of DNA is that they run antiparallel. Genetic code, it is read from 5' to 3', or, well, it runs these strands from 5' to 3' or 3' to 5'. And what this means is like you have phosphate groups at the fifth and third carbon of these deoxyribose. And then they form like a ladder going down. So that's where you have your phosphate groups. So then, if one of the strands runs from 5' to 3', the other one will have to run in the opposite direction. So here we have an example. So you start with the 5' with the C, and then you end up with the T and the 3'. So the one that is going to be anti-parallel will start here, 3', and then end up here with the 5'. And let's base pair it. So what will follow here? G, so that's the arrangement. So then depending on the order of these base pairs is what type of protein you will make or what type of function you will have. So then the two strands are complementary and then can act for a synthesis of a new strand. So if, let's say, if I want to make a new strand and I separate them this, and then I make a copy of this in this direction, so my new strand will be what? Same as that, right? So this will be my new strand. And then if I do one that goes in this direction, then will be the same as this other one, right? G, C, A, T, C, G, G, T, T. So this will be another new strand. So the enzyme that does this work is the DNA polymerase. So here you can see this structure of the DNA. I mean, I know that you're familiar with this. Ribose here, anti-parallel, 5', 3', 3', 5', nucleotide bases toward the center, forming a strong in a ladder, 2 hydrogen bonds between T's and A's, 3 between G's and C's. And then phosphate groups at 5 and 3' end, binding all these nucleotide bases. So then when you are doing this replication with the DNA polymerase, you use several enzymes. First, you use one that basically uncoils the supercoil, which is the topoisomerase or gyrase. And then once you uncoil the supercoil, you use another enzyme to separate the two strands. This is the helicase. And then the helicase, what it's going to do is make a replication fork. So if you have here your two joined strands like this, your helicase will separate them like this. So you can start adding your nucleotides. So this area, it is called the replication fork. So here we have the parental strands, 5' N in this strand, 3' N, and then 3' N in here, 5' N in here. You use your topoisomerase, uncoil the supercoil, and then you separate with this helicase, the helix. So you end up with separated the two strands of the parental DNA. And then this will allow, as you keep going, separating, the replication of both. So you can add your nucleotides, and then you can have the enzymes that replicate these nucleotides. And something that happens in this process is that you have to have like a starting point. So this is when you make a primer of RNA to start. And then your DNA polymerase can start adding these nucleotides. So when it comes to DNA polymerase, it's an enzyme that elongates your strands from 5' to 3'. And then you have this primase enzyme that adds the RNA primer. And then the leading strand will replicate continuously while the lagging strand will be disrupted every time as you keep opening your two strands. And then you form little segments of DNA that is called the Okazaki fragments. And then you use an enzyme that is called ligase to put them together. So, let's see if it has, yeah, you will have that. Okay, so here is a list of the enzymes and their function. So gyrase, I already told you, relaxes the supercoiling ahead of the replication fork. Ligase separates these two strands by getting rid of these covalent bonds. And then polymerases synthesize the DNA. And some of these polymerases, they have a proofreading ability. So let's say that when you are replicating, you put here a T instead of a C, and here a G. So this is a mutation. So then the DNA polymerase, as it's adding them, you will say, oh, wait a minute, I'm misspelling. So I will change it to the correct order. Now, some polymerases are a little bit sloppy. They have this proofreading, but they're not perfect, and they end up adding mutations. Now, we have other enzymes like endonucleases that cuts the DNA backbone in a strand of DNA to help facilitate repair and insertions. So then you can have an endonuclease helping to repair. And then you have an exonuclease that from an exposed end of DNA, so this is in the backbone, this is in the exposed end of DNA, it will facilitate the repair. And then in the newly made DNA, you have an enzyme that is called a metallase that adds metal groups. And these metal groups basically shut down the expression of the genes when this DNA is made right away. And then we have this enzyme photolyase that uses this visible light to separate any UV-induced pyrimidine dimers. So, who remembers what are the purines and pyrimidines? So, will
Pure
be
as gold.
or pure as gold, yes, as well. So another way is pyrimidines has the short sequence or the short structure, which will be how many rings of nitrogenous? One. And then purines has the longer structure. So adenine, one. And then pyrimidines. We have thymine, what else? Cytosine and uracil. So you shouldn't have pyrimidine dimers. That is formed frequently with UV light exposure, thymine dimers. So this photolysis helps to prevent that to happen. And again, like any other enzymes, it's not foolproof, but it helps to have it functional to prevent having these dimers of pyrimidine. And then we have an enzyme that it is related to transcription. So when you make an RNA copy, which is derived from actually the coding of the DNA, of course, you remove intervening sequences, which are the introns, and then you put together the part of the DNA that is expressed, that is the exons, and then you can make your protein. So that is done by these ribozymes. And then we have this RNA polymerase that makes a copy of RNA from a DNA. And then we have SNRPs that are RNA protein complexes that remove the introns and splices exons together, similar to the ribozymes. And then topo, isomerase or gyrase is relaxing the sucralox. And then transposase. This is an enzyme that cuts DNA backbone and then leave sticky ends so that a transposable element can bind to these sticky ends. Like you can have two ways to cut DNA. For instance, you can cut it this way, and then you can have one segment like that, very straight, or you can create a sticky end. So for instance, you can cut it this way. So you end up with this piece that allows for insertion of this, and then you create this piece that allows for insertion of this. That's a sticky end. So you can introduce there a transposable element. So here is a summary of the events of the DNA replication at the fork. So you have your enzymes that unwinds this double helix, and then you create this replication fork. You will add some proteins that stabilizes these strands so that they will be much easier accessible. And then your polymerase will replicate your DNA, and then the leading strand, it will be synthesized continuously from the primer that it was made before by the primase enzyme, which is this. And then the lagging strand will have to wait for this to open to keep having nucleotides exposed so that you can make tiny pieces of DNA that are called the Okazaki fragments. Once you have made 2 Okazaki fragments and the DNA polymerase is done by making copy of one of these segments, then you will have your ligase, which is going to bind the Okazaki fragment. So this is the lagging strand, this is the leading strand. So this happens because you will have the 5' to 3' reading by this DNA polymerase. Any questions? Now, there's a lot of energy consumed when you make copies of DNA. So you use ATP, and then you have to add these phosphate groups to the nascent strands of DNA, and then you end up with losing 2 phosphate groups. So here is adding new nucleotides to these strands, the template strands. So again, you lose 2 phosphate groups, and then you end up just adding 1 phosphate group, and your previous nucleotide that was having 3 phosphate groups now end up with only 1. So most of DNA, of course, has to be replicated bidirectionally. I mean, it doesn't make sense that only to open up the 2 strands to make 1 copy of one of the strands and not the other one. And the purpose is just for the offspring to receive 2 copies of the DNA. Well, 1 copy of the DNA, and then you end up with these daughter cells that hopefully receives this DNA without mutation. So here we have an example of E. coli being replicated. So this is a replication fork, and one is the parental strand, the other one will be the new strand. How would you know? Well, in this case, it's hard to tell which one will be the new, which one will be the old, unless you're following it with live imaging. But in the case of, if you're investigating, trying to see, okay, where is my new strand? Well, the one that is methylated will be your new strand. So that's DNA replication. Now let's talk about RNA and protein synthesis. So RNA, of course, stands for ribonucleic acid, and then typically it's single strand, linear, and in this case you have a ribose sugar instead of the deoxyribose sugar. So you lose this hydroxyl, and then you have uracil instead of thymine. We have 3 types of RNA: rRNA, ribosomal, tRNA or transfer, and mRNA. These 2, well, all of the 3 participate in the process of translation in a way. And then transfer RNA is mainly acting on the cytoplasm, bringing the amino acids into the ribosomes so that you can have amino acids to be added into these polypeptide chains as they are created. And then ribosomal RNA is part of these ribosomes. It is created together with this messenger RNA within the nucleus of eukaryotic cells but it is found, the three of them, in the case of prokaryotes in the cytoplasm. And then the messenger RNA itself is the one that carries this information from the DNA to the ribosomes. So for transcription in prokaryotes, so you have this messenger RNA that it is basically complementary to the DNA template. So now let's change this to, let's say you are making a copy of messenger RNA instead of, you're transcribing instead of a copy of DNA. So let's say that this is your DNA, and this is going to be one of your RNA strands. Let me put it here. And then here you have an RNA strand. Tell me which ones I'm supposed to substitute here.
The T's are supposed to be U's.
Mm-hmm. And in this case, for this RNA, it will be read in groups of 3. So you will have a codon. Which one is the codon that makes methionine? Do you remember? The starting point.
It's AUG.
AUG. So let's say that I want to make here a starting codon. What that I supposed to have? Or well, let's do it here. So that will be, has to be a G, and this one has to be what? C, and then G, and this will be so AUG, this direction. So AUG, this will be methionine. So this will be the codon for methionine. So that's the initiation. So you start transcribing in the starting point, and then your RNA polymerase will bind to this AUG, and that is part of this promoter sequence in your DNA. So after you initiate this transcription, this transcription, you will start elongating. Elongating happens from 5' to 3', so in that direction. And then you will keep adding your ribonucleotides. And only one of the 2 strands is transcribed. So in this case, I wanted to give you that example in there, but it's actually, when it comes to transcription, this one won't exist, right? So this one is transcribed, so you make the messenger RNA from 5' to 3', you read it. And then you end up with a stop codon that is when you reach the terminator sequence. So you have promoter, and then you have an intermediate sequence that you elongate, and then you have a terminator sequence. So here we have the example. So you have initiation, your RNA binds to the promoter region, and then you start elongating. And then as you are elongating, of course, you are making this RNA longer, longer, longer. It's messenger RNA. You reach the terminator, and then you excise your RNA, and then that stops the transcription process. So this is the strand that is being transcribed, not both of them. And then you add uracils instead of thymic. And then here, multiple pieces are made of messenger RNA out of this DNA. And then translation, it is making your actual proteins. So again, we read this sequence of the messenger RNA in groups of 3 nucleotides. Those are the codons. And for a particular amino acid, we have 64 different codons. 61 of them are sense codons that encode the 20 amino acids. Starting codon will be AUG, and then we have 3 stop codons. And that will be UAA, UGA, and I don't remember the other one. But anyway, one thing about this codon is that the last nucleotide base, the third base, is wobbly. So you can change it, and then that still codes for the same amino acid. So here we have it. Oh, and here are the 3. So the 3 stops. So we have here in this row UCAG, and then in this column UCAG. And just like if it was a Punnett square, you combine them to make the different types of amino acids. In this part, in this little segment, you have UCAG, 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 because this will be the one that it is found in the third base. So this and this will be for the second, first and second, the combinations. So everything here will start with UU, and then everything here UC, UA, UG, etc. And then the third base will be given by this. So then anything in here for this one will be like these 2 will be for phenylalanine, 3 U's, 2 U's and 1 C, etc. Starting is always AUG, the stop is UAA, UAG, and UGA. So then because of that, it helps. Thankfully, we have a third base that, for the most part, the mutations that you may have will be silent. So you can have a change in the third amino acid, and you may have an amino acid that it is the same, or maybe it's not the same amino acid, but it's an amino acid that, functionally speaking, it can be doing the same because it has the same charge and does the same function in the protein. So first position is here in the column, second position is in the row, third position is in the right column. So anytime that you have a change in amino acid because you have mutation in your DNA, that gives you this code, UAA, UAG, or UGA. It codes for nonsense codon, which is a stop codon. So let's see. So we have here this UAA, UAG, UGA. So let's say that you have a lysine, which is this, in this case, for 3As, or you have AAG. If you have a mutation that changes this A for a U, you will end up with a stop codon. If you have a mutation that changes this A for a U, you will also have a stop codon. So within all of these codes, there will be some codons that are more subject to give you stop codon and nonsense than others. It's hard to see it just plainly like that, but if you go and look at it at the codon, you may say, well, this one, this amino acid, if you have substitution, you will end up with a truncated protein. And let's say that it can be a protein that codes for a tumor suppressor gene, something that keeps you free of cancer. If you have a mutation in these genes, you don't code for this suppression protein, tumor suppressor gene protein, and then you end up with cancer. So there are certain amino acids that are more important than others. And then the codons, they're supposed to be read sequentially. Now, for tRNA, tRNA is the one that brings these amino acids attached to them, and it does so because it has in one part an anticodon that basically is the complementary sequence to the codon that you have in your messenger RNA, and it's supposed to bind the specific peptides. And then once you have these specific peptides added into this nascent polypeptide chain, you form peptide bonds. Now within the ribosomes, we have two subunits, a ribosomal subunit that is large and then the other one that is small, that when they come together, they form a complex, and then this complex is ready to read this messenger RNA, starting with the start codon, which is AUG, and you have your first amino acid brought by this transfer RNA, which has this complementary UAC, which basically is the complementary sequence for this codon. So that's why it's called anticodon. So within this region, which looks like if it was a cross, this part has the anticodon, and in this part is where you bind your amino acids. And then internally, in your large part or the large subunit of your RNA, you have an exit site, a peptide site, and an amino site. So the amino enters in this A site, then it is the amino acid enters into this A site, and then it gets transferred into this P site, where you form the peptide bond as you bring the second amino acid, and then you have the exit site. And then this one is translated from 3' to 5'. So that's the initiation of translation, and that just as for the transcription, you'll have more, in this case, amino acids coming with the anticodons, so you will elongate your messenger RNA as you make, your proteins as you make the entry of more of these amino acids. So you keep adding them, and then you have a complete protein, you stop the translation, and then you release your polypeptides. It is not said in your book, but typically you end up at the end with a poly-A tail, so you add a bunch of A's, adenosine. So when it comes to bacteria, the translation begins before the transcription is complete. So you don't have to wait. It's more efficient. In the case of messenger RNA in eukaryotes, you have to have transcription to stop because the messenger RNA has to exit the nucleus in order to have translation in the cytoplasm. Because we don't have in the nucleus the ribosomes, we have it in the cytoplasm. So you have to not only release the messenger RNA from the transcription area, but also in that case for eukaryotes, you have to process your messenger RNA. You have to cut the introns, join the exons, and then once it's ready, the message, you ended up putting it into the cytoplasm. So here is then the simultaneous transcription and translation. So you have your RNA polymerase making messenger RNA, then your ribosomes come as polyribosomes in many cases, like a chain of ribosomes, and then you start translating. And then that you can see it in here. So this is a longer ribosomes chain. So in eukaryotes, then you have these exons where you code for proteins and introns that do not code for proteins. And then your small nuclear ribonucleoproteins, or SNRPs, removes these introns and then put these exons together. So this is called splicing. So here we have an example. In DNA, you have exons, introns. Introns stands for intervening sequences. These were, I don't remember which year were discovered, but people thought about, oh, what a waste of DNA. Well, it's not a waste. This is to control the proteins that you want to make. It's like, okay, well, someone likes to bake a lot, and he buys a bunch of flour, eggs, butter, sugar, and then we'll be like, wow, you're a hoarder, you're bringing a lot of things and you're not going to use it. Well, out of that 50-pound or 25-pound bag of flour, dozens of eggs and butter and milk, you can make cookies, you can make cakes, muffins, scones, you name it. So it's not a waste. We have these to control what exons are expressed. Like if we number them, you have 1, 2, 3 exon, and then you have these 2 introns. If you want, you can make a combination of exons 1 and 2, or 2 and 3 only, or you can remove these introns and join all these 3, so you can have 1, 2, 3, or you can have 1 and 3. So it depends on what do you remove. And this process is splicing. So removal of introns and bring them together, these exons. And then after you have a complete messenger RNA, exit the nucleus through its nuclear pore, and then you start forming your proteins. Okay, so what is the role of the promoter?
Transcription. Sequence.
So you activate your transcription. Terminator.
The sequence that ends the transcription.
And the the role of messenger RNA in transcription? Brings the message, right, about what you're supposed to make. And then what is the difference between messenger RNA production in eukaryotes and prokaryotes?
It is produced in the nucleus, and then in prokaryotes it's
So you don't have nucleus in the prokaryote, so you have to make it in your cytoplasm. So now we're going to see what is an operon and how do you regulate gene expression in bacteria, and what is the post-transcriptional regulation of gene expression. So when it comes to genetics, the genes can be expressed at a particular time or not. There are certain genes that are always expressed. These are constitutive genes, and they do not appear to be regulated because they're always on. And other genes are inducible, so they are not always on, but they are expressed when they are needed. And other ones are repressible. So maybe you want to not express those genes, and then some of these genes can be regulated when you have a specific molecules. So this is catabolite repression. So in pre-transcriptional control, before you make a protein, you might want to have repression of these genes because that protein might not be needed all the time. So in this case, you use repressors. So repressors are proteins that block transcription. They can be bound to specific regions of DNA, blocking the RNA polymerase for coming in and transcribing these molecules, or these genes, sorry. So the default position of a repressible gene is on. So genes that are always on can be repressed. And then induction will be expressing genes that are not expressed. So this is done by an inducer. So the default position of inducible genes is off. There are some times in human behavior, guys, people who is very verbal. They tell the truth very bluntly. So in this case, the repressible gene is always on. But there might be coming someone who can make this person to reason and say, you know what, try to be less offensive. You can still say the truth, but be tactful. And now, in induction, it's like some of us, we are shy, and we always let the other ones to express themselves. But then there's someone who's more bolder than us and tell us, you know what, tell what you need to say. It's not wrong to say it. So in this case, you are kind of always off, but if you have an inducer, someone that tells you, well, come on, jump in, tell, then you have some gain in there. Now, within genes, specific genes are known as operons, like a segment of genes. We have a region that is called the promoter region. So this is where you start the transcription by the RNA polymerase. And then this promoter will be next to a region that is called the operator. So the operator, it is this segment where you have sequences that controls the transcription of these structured genes. And then the operon is both the operator and the promoter sites and the structured genes that they control. So we have two types of operons, one that it is inducible, and in this case for inducible, you have structural genes that are not expressed all the time. And you have to have an inducer to induce this operon. So for instance, the lac operon in E. coli. In the lac operon, you have three structural genes, C, Y, and A. This codes for enzymes that allows E. coli to utilize lactose. And then next to this, you have a promoter region where the RNA polymerase will start transcribing the messenger RNA to make the proteins that come from these three structured genes. And then you have an operator that is basically where you can stop or go the signal. So when you don't have lactose, the repressor gene that it is upstream of these sequences, that it is coded by an I gene, will be binding into the operator, preventing transcription, because RNA polymerase cannot bind there. Now, when the E. coli cell finds in the environment lactose, then these proteins that are in the cytoplasm of E. coli can change lactose molecule into allolactose, that is an inducer, and then this inducer will bind to the repressor, and the repressor can no longer bind to the operator, and then you have transcription. So here we have the lac operon. So here we have the I gene that codes for the inducer. And then here we have the promoter region. And then here we have the operator. And then here is Z, Y, and A. This one codes for an enzyme that is called beta-galactosidase. This one for an enzyme that is called the permease enzyme. And this one for an enzyme that is an acetylase, transacetylase enzyme. So when you don't have lactose, there is no purpose to express these genes that are used for lactose. So in this case, the repressor will be binding to the operator so that you don't have this RNA polymerase binding. So here we have an active repressor protein coming from the I gene. So since you have a physical contact of this repressor into the DNA, polymerase cannot express and cannot actually transcribe these genes to be expressed. So then this operon is off because you don't make these proteins. Now, when you have lactose available and you convert it into allolactose, then this allolactose can bind into this repressor that it was inactive, and now it can become expressed, this operon, because this one that was binding in here, once you bind this allolactose, it will remove from the operator side, and then it becomes inactive, and then your RNA polymerase can transcribe this so you can make this messenger RNA so you can have beta-galactosidase, permease, and transacetylase. So in this case, this operon will be on because you are transcribing, and then you translate these proteins. So that's an example of inducible operon. Next time we will see the repressible operons.